Good evening, everyone. Can you hear me? Oh, need to be just very neat. I'm Narinder Sufi. I'm a Deputy State Librarian here, and I have the pleasure to welcome you all today to our night at the State Library. Thank you very much. Before we introduce, Alex is going to introduce our speaker, tonight's speaker. I would also like to thank the foundation, the Library Foundation, California State Library Foundation, for supporting this event and their continuation of that support. Thank you very much. Our next night at the State Library will be on May 9th? May 9th. And that will feature John Clickenbeard, a geologist with CGS. And the talk will be about the history of gold and mining in California. So I hope you're able to join us again that time on May 9th. Thank you very much. Alex is going to introduce tonight's speaker. Thank you.
Good evening, everybody. I am very happy and proud to introduce our speaker tonight. We selected the speaker because it is Women's History Month, and so we were looking for someone who would bring a unique perspective on a unique field. Dr. Judith Connor has been a marine biologist for quite a while, and she has taught a class in Antarctica. She's lived in an underwater habitat. I believe it gone to all 7 continents. All 7 continents. I've known her for quite a while and have really enjoyed our opportunities. We've gone snorkeling together. We've gone scuba diving together. Haven't gone on all of the adventures, but have been there for a handful. And I'm just really, really excited to share this with you today. So thank you very much.
Thank you, Alex. And boy, it's wonderful to be here. You know, it was raining on the coast when I left Monterey this morning. And this afternoon, the sky opened up here, and Sacramento is such a beautiful place. And if you haven't been to the coast lately, well, between Sacramento and Monterey, it is green, it is beautiful. Come and get it while it's still wet. Just love it. So it's very exciting to be here and talk about changes in marine science during my career, and also changes in the tools that we use to study the world ocean. I have to tell you, when I was a kid, I didn't dream of being a scientist, much less a marine scientist. I'm the 7th of 10 kids. My father went to college for a semester. My mother graduated from 6th grade, and so I didn't have role models to even consider what a woman might do in the new world that we're living in now. So very exciting to be able to share with you my adventures in marine science. Started at the University of Maryland. I had to take 1 science class to graduate. So freshman year I took botany, and I discovered I loved botany. I wanted to be a botany major, but they said you had to take chemistry and physics. So I thought, I don't know. But I took chemistry. I loved chemistry. I took physics. I loved physics. So I ended up with a double major in biochemistry and botany. And one of the tools that really changed my life was a microtome knife. Now, I know I look like I must have been a nice girl. What is she doing with a knife? But it was a class that I took, and they gave each of us this sharp knife, and they also gave us a strop, so we had to keep them really sharp. And the way a microtome works is you put your sharp knife into this machine. You can embed like a stem or a leaf in wax, and then you can slice it 1 cell layer thick and go through the whole plant, just like you would with an MRI, but this is the old-fashioned way of doing it. And I got really good at it. I guess being forbidden to play with knives as a child, I really wanted to do something with this. And ironically, that led me to an interview at the Smithsonian Institution in Washington, D.C. And so the first day I went in for this interview, I met Walter Adey. He was a paleobotanist. He studied coralline algae on reefs, and from the Arctic all the way down to the tropics. He was studying these weird pink encrusting algae. Here you can see some pictures. So here, this whole rock-like structure is pink coralline algae. I think Linnaeus, when he first described it, he thought it was an animal like a coral. And they do have a really important function in coral reefs of holding the reef together. The coral animals are much more fragile than the coralline algae. So what Walter wanted was somebody who would microtome through these crusts. You have to decalcify them and embed them in wax, but then layer one cell layer after another until he could actually see the little holes that you can just barely see on the surface of the coralline algae actually form these reproductive chambers, and that's where the eggs and sperm of these coralline algae are formed. I mean, it's remarkable that you have reproduction coming out of pink rocks. But anyway, the first week I was there, you know, I said to Walter, you don't have to pay me. Keep advertising, find somebody else to take this job because I'm moving to California. And the first day I was there, I worked from 8 a.m. until 8 p.m. And he knew he wanted me to stay. And so at the end of the week, he said to me, I'm planning an expedition. It's a 3-year expedition. And we're starting here in Washington, D.C., and we'll go down and then out through the Caribbean island chain. And if we have enough time, we'll go over to Africa and go up the coast of Africa. And I said, oh, what about my sweetheart? And he said, well, is he good with his hands? And I said, is he good? Well, the reason why Walter wanted someone who was good with his hands because we had to build the boat first. So this was Walter's backyard, and we built this trimaran in his backyard and then hauled it with a big truck over to the Potomac River, Alexandria, finished putting it all together, and then just as the Potomac River was starting to ice up, we took off. And we were still working on the boat as we worked our way down the intercoastal waterway to Miami. But eventually, we got the sails up, we got the engine working, and here you can see the back of the ship, of the boat. It was a 41-foot trimaran. On a good day, there were 6 of us working on it. On a bad day, there were 9 of us, and there was not enough room for 9 of us. But Walter was great in terms of he expected each of us to help with the tools, just like helping with the building of the boat and doing the microtoming. Each of us had a certain motor we had to take care of. So my sweetheart, he had the main engine, the diesel engine on the boat, and at one point I was in charge of the generator, and another part I was in charge of the tank that we used to fill up the scuba tanks, the compressor. And so, you know, never having taken any machinery courses, you know, he would have books and you'd read them or you'd ask for help, but essentially he was hands-off. He expected each of us to do our work on our own. And so the interesting thing was that we'd also do geologic drilling because he was interested in looking at the organization of coral reefs down through the millennia. And so this looks easy, geologic drilling if you're drilling in a driveway, but what we would do is build a big tabletop on top of a reef, and these kinds of algal ridges only develop under really wave-washed reef activities. It's when the reef grows up to the sea surface and the animals can't take that kind of wave action, but the coralline algae can, and they just keep building up as long as they get wave splash to keep them wet. So we did a lot of geologic drilling in the worst possible places that you can That's why I wear colored hose. All the times those waves knocked me over on top of the coral. And here you can see, after we went down, 30 feet through the reef, we would lay out the cylinders that we had drilled out, the geologic drill, and then we'd take those and identify what we could of the corals, the coral and algae, and then do carbon dating to find out how old the reef was over time. And we used aerial photography for mapping. So mapping is still a very popular thing to do on the sea floor. In our case, we used cameras. So we had a Rolly and a Hasselblad and a Nikonos for underwater. And the aerial photographs, we would rent an airplane with high wings, and it had to be high wings because we'd take the doors off, and then we'd strap my sweetheart in, and they would fly out over the reefs, and he would lean out the window, and they'd turn the plane on its side, and he'd take these pictures of the reefs. And here you can see, okay, there's Pat and me in the water with our rubber dinghy when he's flying over. And here's an example of what we would do is we would project those slides. We were like developing slides inside sleeping bags in the boat. And we would project the slides and then project them onto Mylar so that we could outline where the reef was and where the color changes was. And then we'd take the Mylar out in the reef when we went snorkeling to identify all the algae and fishes and corals and what was happening. So we got these really detailed maps of what was going on different Caribbean islands. And I can remember the first night we were heading down the Intercoastal Waterway, we were sitting around the table in the boat, and Walter said to one guy on the boat, okay, you're gonna do the corals, and you are going to do the fishes, and Martinez, you are gonna do the algae. Well, he said, Connor, you're gonna do the algae. I went, I don't wanna do the algae, I wanna do something cool. Give me the fish, give me the corals. He went, no, you have a botany degree, you get the algae. And so I got brown algae, like these little sargassums and turbinarias, not nearly as glamorous as our kelp here in California, but pretty diverse, pretty cool. And then also some really beautiful greens, like this little wine glass Eudodia, and then these flaming red, like Halimenia there. And so, if you do anything every day for 3 years, you become an expert. You can't help it. And so I spent many hours on my hands and knees looking at, a 1/4 meter square, an area about this big, that I would identify everything growing in that. And in a 1/4 meter square on these reef flats, you can get 100 species of little tiny algae in 1/4 meter square. So it would have been tedious, except for I was just enthralled with it by that point. Very, very exciting work. And the fact of having that much experience every day out on the reef, it really opened other opportunities. So I had many invitations to join research expeditions, initially in the Caribbean. And here, the women scientists you see on this cruise, on the research vessel Seward Johnson out of Harbor Branch in Florida. So Harbor Branch Oceanographic Institution, they had this fabulous ship. And on the back deck of the ship, they had a sub. You can call it a human-occupied sub. I like to call it a woman submersible. So that's me sitting next to the sub-pilot. And it's just really fun because you have almost 360-degree view. And to save the batteries, because you're detached from the ship, you go down in the darkness, and then when you get to the seafloor, you turn on the lights, and it's fabulous. I mean, it's just so amazing. So in addition to the microtome and scuba and snorkeling and cameras and airplanes, now I was using a woman submersible, and it gave me a chance to go to really wonderful places. Like here you can see a picture of Cocos Island. It looks like Treasure Island. It's this mountaintop that comes out of the ocean here, 360 miles off the coast of Costa Rica. One mountain that just comes up. And then also we took the Seward Johnson and the sub here to the Galapagos Island, which I liked very much because my scuba friends down there in the deep water were, they were seaweed-eating iguanas. It was so cool to be underwater and to have something in there that also appreciated the seaweed very much. And Cocos was cool. I actually got a chance to go to Cocos. I was not originally on the list of people to go, but there was a chemist who was going to go on the research cruise, and he had just learned scuba diving, and I said, well, you're going to love Cocos because they have so many hammerheads. The hammerheads always go there to mate. And sure enough, when we got there, there were hundreds of hammerheads. Anyway, he dropped off. He was not interested. I really told him, you shouldn't, oh, maybe you should worry. I never worry because I always think of them as man-eating. And other places that I got to go with additional expeditions, some sponsored by pharmaceutical companies, included studying the waters around Spain, particularly in the Mediterranean, and then out the Canary Islands off of Morocco, off of North Africa, and then out using the Turkish National Research Vessel around the Aegean. That was sort of exciting because when you got in the water, I mean, it was so exotic for me. You know, the call to prayer in the morning, I couldn't read any of the signs in the cities. I couldn't understand the language. Every time I went to a new country like Spain or certainly Turkey, I always learned enough to say, help. So that if I got to the surface and there was no boat and I couldn't find my buddy, I could yell for help. So I don't even remember them now, but if I went back again, I would relearn how to say help. And then later, I was invited to go to Namibia, and here you can see Namibia is just above South Africa, down here on the tip of Africa, and the whole country is sort of like this giant, amazing desert. The desert dunes are as big as the mountains here, the Sierras. I mean, they're huge, and they just look like sand dunes, and we had a desert storm blow in when I was working there. And desert storms are amazing because the wind will be whipping 50 or 60 miles an hour and the sand is moving. And that's why I look so youthful, all that sand. But it was fantastic. The water was very, very dark. Namibia was where I got my first dry suit. So the water was colder than Monterey Bay. It was actually more nutrient rich with upwelling. So you know how Monterey Bay, sometimes it can get thick with phytoplankton. It looks like pea soup. It's not so fun for scuba diving. But here you had this nutrient rich water that was even thicker. So when you got in to do a dive, you'd say to your buddy, stay close, and then you'd never see him again. Even if he was 2 feet away, you would not see him. And then if something bumps into you while you're diving, you'd wonder, is that a white shark? Or is it a penguin? Or is it my dive buddy? You know, it was just really crazy. And there were diamond mines everywhere. And so the government really wanted to make sure that if you were scuba diving, you weren't doing something like eating diamonds off the seafloor because they wanted to keep all the diamonds under control. It was one of the most challenging places that I have been diving, and just quite an experience to be there. So with that kind of field experience, I thought, you know, I really want to do my own research to really look out and have my own, you know, I can be part of the team, but I also want to really discover things on my own. And so I moved from the East Coast to California, finally, and went to the University of California at Berkeley, and it was a wonderful experience. I think I was there like 2 weeks when I was invited to go back to the Caribbean and be part of a group that was studying drift algae in a submarine canyon off of St. Croix in the US Virgin Islands. So the hydrolab, which you can see here in the background, and you can see the air bubbles escaping here. Hydrolab was about 50 feet deep, and they had a, if you look at the window of the hydrolab, they only have one window and it faces out. So this is the inside of the hydrolab. 4 of us were in there at one time, and there were 2 different teams. So one team was down for a week, and those of us that were on the surface brought things down and talked to them about what we were going to do from the shoreline. And then the second week, I was one of the 4 members of the team, 3 men and me, and I didn't know any of them. And it was pretty confining. So that's the inside, and somebody has to stay awake all the time so that you can keep on the radio to the people on the surface that everything's going okay, the oxygen levels are fine. They had sort of like a coat rack. So if you can imagine, the door was in the bottom. The air came pumped down from the surface, and so the air would then burp out of this door that was on the bottom. And if you can imagine, if you wanted to get your tank off the coat rack in the distance, what you would do is you'd get on your mask, you'd settle down, and you'd look at where's the coat rack, and then you'd stand up. You'd be in air at the door, and then you'd be in water, and then you'd be in air, and then you'd take a deep breath, and swim over the coat rack, and get the regulator in your mouth, and then put on your, and then you could swim out there all day because you're saturated. You can't go to the surface, but you can go down to 100 feet, you can go to 150 feet down the canyon. It was amazing. It was amazing, even living with the challenge of no privacy whatsoever. Thank goodness I was one of 10 kids, so I knew how to deal with that. So, and that's what it was like, sleeping when the other people in the hydrolab let you get a little sleep. So while I was at that stage of being invited on expeditions and having exciting adventures, I had also gone to Honduras and Guatemala and Belize and down to Venezuela, and a short visit to Panama, and I was really curious about Panama. And so going forward, I actually chose the Smithsonian Tropical Research Lab in Panama as the site for my PhD research. And it was very interesting. I was looking at reproduction in the small seaweeds that grew on the reef flat at the Galeta lab. And here you can see the reef flat, and this was during the calm season when there's not a lot of waves washing over. And so a lot of the seaweeds were dying back, and I was interested in when do they go reproductive, or is it all asexual? Do they have sexual reproduction going on the reef flat or not? And as you can see, I also was doing some reproduction on my own. That's my son on my backpack, who was born in Panama. And when he was born, I didn't speak any Spanish. And boy, I learned quickly, you cannot survive as a mother without a lot of help. And so I learned, I think my first full sentence was, ¿Puede usted ayudarme con mi niño? Can you help me with my kid? Or where do you find the diapers? Just basic things that when you work in an all-male lab, nobody seems to know where the diapers are. But wonderful experience being there for 2 years, and then back to Berkeley, finished up my PhD, and I became a postdoctoral scholar and came down to the Monterey Peninsula. So here's Monterey Bay. Of course, Berkeley and San Francisco's way up here, and Sacramento's way over here, but came to the Monterey Peninsula, and I was a postdoc at Hopkins Marine Station of Stanford University, which is right next door to where the Monterey Bay Aquarium is now. And this was the view from my office at Hopkins Marine Station. So there I was looking more at the biomechanics of seaweeds and how intertidal rocks provide habitat for different seaweeds, and what are they doing in terms of reproduction. And what was really fun was to become a lecturer at Stanford and have the opportunity to teach a class in marine biology for 10 years on the side. It wasn't my full-time job, it was my side job. So I'd take vacation from wherever I was working or whatever else I was doing and teach, and just such great students. We collected wetsuits so that everybody could get their head in the water. I mean, how can you study marine biology from land? It's crazy. So the whole purpose, our tools there were our creativity and collaboration, our snorkeling gear, including the wetsuits, microscopes, and each of the students had to pick a weed. It was like adopt a weed and bring together the information that we already know about that seaweed, but add to our knowledge. And so these students, they're so competitive. And I kept having to take them aside going, this is a collaborative project. I know you're an A student normally, but you're failing here. And they ended up being just really great collaborators. And just for an example, this was James Lopez. You see him looking through the scope here. And one of his projects was to describe the life history of Macrocystis, the giant kelp. And here you can see the sporophyte produces these spores. They're like kidney beans with flagella, and the female settles down, and then the male, that's the male, that's the female. The male releases sperm. It's attracted to this egg cell on the gametophyte, grows up to be the sporophyte, which then is the plant everybody else knows out there in Monterey Bay. And so well, I love cartoons. What can I say? Anyway, I thought the whole idea of these students making animations to explain complex things, I loved those students, really had a good time. And being at Hopkins Marine Station, when two of the professors at Stanford were invited to be part of a National Science Foundation class in Antarctica, which was really based on, it was an international biology class to bring new PhDs from all around the world to Antarctica so that they could start considering the kind of research they might want to do if they wanted to work out of Antarctica. So essentially we were based in McMurdo, and we did not come by ship, we came by air. So it's an 8-hour flight from Christchurch, New Zealand, down to McMurdo. And the first time that we were heading down there, we got all the way down there, and they were having a whiteout storm, and they said we couldn't land. They couldn't find the runway, and they sent us 8 hours back. Oh, it was miserable. And then we had to wait for a couple of days while reports were coming back. Finally, the storm cleared. 8 hours, got dropped off there. It was interesting because the tools are very different in the sense that we had to all learn to do snowmobiles so that you could get out of where the town was and get out to field stations. And we also used helicopters to get out to more remote places where we were going to dive. And just certainly some beautiful, beautiful vistas down there. Of course, my work, in addition to giving lectures, was also to be the scientific diver. And I got a new dry suit for that trip. And the clothes you wear in this dry suit, it's sort of like, what do you call that, like a snowmobile outfit. And then you put your dry suit over it, and you have, oh my gosh, you have so much clothing on that I had to wear like 60 pounds of lead in order to get down at all. And talk about getting down, it was a trip because the ice there was 20 feet thick. So, and the ice hole, you know, they have an auger, and they open up a hole that's just big enough for one person and their scuba tanks to go down at a time. And so, you know, I got in the water and made sure my mask was okay. In fact, your whole face was covered. You had a gorilla mask around your mask, and so your forehead's covered, everything's covered except your lips and your regulators in your lips. And I just, you know, after diving for 45 minutes to an hour, I would come up and your lips are so swollen, I looked like Angelina Jolie. And they laugh. Anyway, diving down, that was incredible. And the cool part was to be able to go and collect a few organisms so the young postdocs that were waiting on the ice at the head of the hole, they could have something to work with in the laboratory. And the underwater scenes were absolutely beautiful. We had sponges that were as big as the chairs you're sitting in. Nemerdian worms, which are like this big in Monterey Bay, they were like this big down there. I know you all love Nemerdian worms. And so following that time at Hopkins Marine Station, I climbed over the fence and also on Monterey Peninsula, took a job at the Monterey Bay Aquarium. And that was so eye-opening for me because it was really about providing science so that artists that work there can develop these fabulous exhibits, and they want everything to be really accurate, which is great. So just working with super talented people, I was helping to develop exhibits, but I also had the opportunity to write books like this Seashore Life on Rocky Coasts, and then some of the other books were Kelp Forests, Jellies, Living Art, the Deep Sea. It was very exciting to come to this great state of California library and find my books in the library. Super cool. And I don't know how many of you have been to the Monterey Bay Aquarium? Hey, love it. What I loved was the first time I took my parents and I had them step here in front of the kelp forest, and they never could get me. They just could not understand what did they do to deserve this daughter who is, swimming with the sharks or whatever. And then when they stood in front of that kelp forest, they got it. It was just really cool. They never doubted me after that. It was very cool. One of my last projects there was taking advantage of this research vessel that was attached to a remotely operated vehicle, or ROV, and the ROV had a television broadcast type camera on it, and it would take video of what you see in the deep sea and send the video back up through the cable to the ship. At the same time, the ship would send the signal, the microwave signal, up to a relay station on Mount Toro, just outside Monterey, and then down to the aquarium. So visitors to the aquarium could see whatever was going on with the ROV with a 3-millisecond delay from the time the image was taken. So, you could conceivably pick up a phone and call out to the ship and say, see that rock over there, would you pick it up because that's got a new tunicate that I want to describe, or something like that. It was just really cool. And so my job was to look at all the video that was coming back from the research vessel from this very new institution that Dave Packard had started. Dave and Lucille Packard paid for the building of the Monterey Bay Aquarium. And then afterwards, Dave Packard wanted to do something to really bring advanced technology to ocean sciences. So he started a sister institution with separate funding and separate vision, separate board, and it was the Monterey Bay Aquarium Research Institute. And so after working on that program, I felt like we could do so much more with video than just showing it one time. And so I left the Monterey Bay Aquarium and joined the Monterey Bay Aquarium Research Institute and moved from the Monterey Peninsula, about 30 miles north, to Moss Landing. Moss Landing is at the head of this deep submarine canyon that starts in about 30 feet of water and gets wider and deeper as it meanders offshore until about, I would say, 40 miles out off of Moss Landing. It opens up on the deep sea floor, the true abyssal plain. And so it's 4,000 meters deep, 13,000 feet out there. So it's the perfect place to be able to study the deep sea with things like ships and remotely operated vehicles without flying to Ecuador and then taking another plane out to the Galapagos Island. And on that trip to the Galapagos, I was away from home for 5 weeks. I mean, it's hard to be away from your family that long. But it was amazing. And so as much as I loved the aquarium and working there with artists, this seemed like a great challenge. And so at the Monterey Bay Aquarium, we have the Western Flyer. It's our big ship. It has bunks for 25. Usually it's 10 of those bunks are for the science or engineering staff, and 15 of the bunks are for the ship's crew and the deckhands and the engine maintenance, you know, the engineers that actually run the ship, and the sub-pilots. And here you can see there's a moon pool in the middle of the ship, and so when you're launching the remotely operated vehicle, this is the Doc Ricketts, when they're launching it, you actually open up the moon pool and drop the ROV right down through the middle of the ship. And then the scientists and the sub-pilots sit in a control room, and they have all these cameras so they can control the ROV. The pilot sits right next to the chief scientist who controls the camera. And so the ROV pilot can see when the scientist is zooming in on a rock or an animal, it's not that the ROV is going to run into the wall, it's the camera zoom. So by sitting close, they can really communicate well and get some really fantastic videos. We also have, in the past few years, acquired the Rachel Carson. So this is, it doesn't have as many bunks. It has 10 bunks, but they're all in one room, so not ideal for long cruises. But here on the back deck, you see a big crane, and this crane will pick up the Ventana, this other remotely operated vehicle, but it launches it over the side of the Carson. And then what you can barely see, but here there are other equipment on the back deck. For example, these AUVs, autonomous underwater vehicles. So when we started the AUV program about 12 years ago, I thought, not in my lifetime, but they actually have done it. It's really amazing. So these AUVs, they're pretty big around, and they're the shortest part of sending them out to sea is you make one that's 5 feet long, but more frequently they're 15 or 20 feet long. So it's not something that even strong women can't carry them around. But we've been able to develop really good AUV programs, including one that has multi-beam sensors that can go out and get really close to the sea floor, and so it gets really high resolution bathymetry data like you see here. So here is the Monterey Canyon, and you can actually, when you go in really close, you can see ripples in the sea floor of the bottom of the canyon. It's unbelievable. And so then you can use these maps that you get from the autonomous vehicle when it comes back home, and you download the data, make these maps, and then decide where you're going to go back with the ROV and really get good images. And now we're outfitting one of these AUVs with camera and sensors so that we'll be able to go out and detect like swarms of krill and also be able to visually identify what animals are in the swarm. So you get more of a sense of what's the zooplankton out there that somebody knows it's out there. You can see it on a lot of these acoustic, but you can't tell what they are for real. And so the combination of acoustic data and the visual is really going to make a big change in the way we study the midwater. But how do we keep track of all these tapes? We have, I think, something like 26,000 hours of videotape from the past 31 years of MBARI, the Monterey Bay Aquarium Research Institute. And we've seen some just far out things like, oh my goodness, this fish that has tubular eyes that point up. Oh, when we posted that on YouTube, it went viral. When I came into work the next day, it had gone around the world and had a million viewings already. And I was only responsible for like 100,000 of them. And then, you know, the comb jellies, these weird, the jetty crab, and this worm. I mean, as soon as we saw that, the scientists were going, oh my gosh, it looks like a butt. And the woman who described it for science, she gave it the Latin name buttworm. So how do we keep track? We have 5 research technicians that work in the video lab and look at every videotape. Even if it's a geology dive, they still identify all the rock formations and all the animals, everything they see. If equipment is put down on the seafloor, they identify where it goes exactly. And whatever the time code on the videotape, it ties in with everything they collect, all the other data. Depth, lat, long, how much oxygen's in the water, nitrates, how much fluorescence. And so, you know, when I first took on that project, I was actually trying to type out the names of all the animals and geological formations, and you couldn't do a one-day dive in 3 days. It just took so long. So we developed this software system, which is pretty amazing, and it makes it really fast and easy that you pick out, are you on the seafloor or are you in the midwater, and you put, you know, what are the animals you expect to see, and then all you have to do is like tap that, and it grabs the time code and it links it up with all the other data. And in fact, all the data from the past, let's see, 27 years of collecting data, it's all available on our website. So high school students, college students, you know, a lot of the graduate students in the Monterey Bay area have used the database, go in, type in, okay, vampire squid. How often have you seen the vampire squid? And you'll get back a spreadsheet that tells you all that data and even gives you images of what the animal or geologic feature looked like. And so the video annotation and reference system, the software is available. It's open source. And so several other big institutions are putting the system in theirs. And here you can also, you can just barely see it, but it says www.mbari.org/VARS, which stands for Video Annotation and Reference System. And then check out the MBARI channel on YouTube, because these research texts are not only beautiful and smart and so collaborative, they're also funny and creative. And so like for Valentine's Day, Kira came up with everything heart-shaped in the deep sea. And then Linda had to do one better, so she did big teeth on Halloween. I mean, they do just such great YouTube videos, so check that out. Don't miss that. And other tools that have been developed, you can just barely see, but here's it's like a water vane. It looks like a weather vane. Essentially, you set that on the sea floor, and it will go into the current. And if the current gets really crazy down on the sea floor, deep in the canyon, it will release this orange football-shaped things, which will go up to the surface and radio home. There's been some big event, and the sediment is really rushing down like an underwater landslide in the submarine canyon. And then also, here you can see Larry Bird, one of the mechanical engineers who I think is genius. And he develops all kinds of things. Scientists come and go, I'd like a fake boulder that I can put on the sea floor, and I'd like to know when it's rolling down, how is it tipping or is it sliding? And so he'll invent these fake boulders with computer systems in them, and then we can go back with the ROV and collect them after 6 months or after we know an event has occurred and we want to see where exactly it has landed. And then this was an example of one of the events where there was a sediment transport event. This is the Monterey Canyon, and you can see, okay, they had moorings that they put out here, and then here was a boulder that they set it down here and it moved that far. And some of the data that they collected at this event, January 15th, 2016, it's more data than has ever been collected on one of these deep sea submarine sediment transfer events. Just super cool. Other tools, here we're now working on a long-range AUV that's much smaller, and one strong woman could pick it up and carry it out and put it in the water. So it's very small, so any instrumentation you put in that has to be low energy and lightweight and compact. And it's capable of the batteries taking it from Monterey Bay all the way across to Hawaii and collecting data all the way across. That's phenomenal. When you think one ship day can cost from $15,000 to $25,000 one day. You know, anytime you can keep your ships in and save all that fuel and all those Dove bars that the people eat in the kitchen, you know, you can save a lot of money. And also here's an in situ ultraviolet spectrophotometer. It's all off-the-shelf equipment. It's a spectrophotometer that gives off ultraviolet light, and when the light bounces off particles in the water, sends back a signal, you can detect how much nitrate is in the water. So that's really important in terms of looking at upwelling, what really feeds the plankton and zooplankton in the bay, but also what's running off the ag fields in Salinas Valley, what's running into Monterey Bay. And it's a lot of interest in freshwater uses as well. And then we have the benthic rover. This is a rover that is about the size of like a Volkswagen, and it actually sits on the seafloor, and it can slowly cruise across the seafloor without stirring up a lot of sediment. It's got these 2 plexiglass chambers in the front, and it can set down the chambers and then track how much oxygen, what's the physiology of the animals that are living in the sediment in that particular area. And it can sit there for a day and then lift up and then slowly move away and do another 2 you know, it can go. And currently, we've really pushed the engineering team to make it so that we don't have to go out every 6 months and change the batteries. And so currently, the benthic rover is able to stay out for a full year in 4,000 meters, 13,000 feet of water. Pretty amazing. The environmental sample processor, oh my God, look how big this thing is. It's a genomics lab in a can. And I just think it's awesome. So this is a can that, you can see it here. If you peek inside it, it's capable of taking in a sip of seawater, and it's got filter disc. It can take in the seawater and filter the seawater so it gets all the particles out of the seawater. There they are, plankton and other little things. And it'll crush the cell walls, extract the DNA with solvents, and then run an assay in real time so you can see what's in the water, whether it's maybe larvae for some important species, or maybe it's harmful algal blooms. And then when you add the reagents, it will develop a picture on the little filter, and you can not only see what species are there that you're checking the DNA for, but also the brightness will tell you what's the quantity of that particular organism, and then send the message back to land so that you don't even have to go out to sea. You can get the message and find out, oh my gosh, we're having a harmful algal bloom. Don't eat any of those mussels or whatever. And you can have them set up near, on a piling. You can put them on a remotely operated vehicle. And right now, we're developing a really small version, small enough that it'll fit inside this long-range AUV. And we've built 3 of these long-range AUVs with the ESP, the small version inside, and just ship them off to University of Hawaii, where they'll be used for the first time this month. So this is what we sort of see as the future tools of marine science. My goodness, it's come a long way from the knife, hasn't it? It's just incredible to work with engineers instead of being the person that's building the ship you're going to be living on for 3 years. Just incredible. So very exciting place, lots of new things coming up, and want to remind you guys that there's a website that's always full of information, and it's Monterey Bay Aquarium Research Institute. And once a year, an open house. This year, it's Saturday, July 21st, from noon until 5 p.m. in Moss Landing. It is so much fun. It is just, you know, the staff, we have tents all over the parking lot. We have lectures going on upstairs in the forum all during the day. And, you know, people bring out their favorite tools, and it's just really, really cool. So I encourage you to think about it, and love to see you in Moss Landing. Any questions? Great. I went to the University of Maryland for my bachelor's, where I got my bachelor's in botany and biochemistry. And I was not a great student. And so after I worked with Walter Ady and I knew I wanted to go to a really good school for graduate school, I went back to the University of Maryland, got my master's and got all A's. And then I applied to 5 top schools and I got into all of them, which was awesome. You know, you can be a jerk in high school and still make it. And that's when I drove across the country in my 52 Chevy pickup and landed in Berkeley, and the car got towed the first day in Berkeley. I loved Berkeley ultimately anyway, but it was cool. Any others? Go ahead. Oh, good, good. How are the trips with Walter funded? The Smithsonian has really generous, or at least back in those days, had really generous travel options. And so everybody I knew that was a curator at the museum was going incredible places. And Walter really wanted to push the limit, and that's why, you know, he had a research vessel that was supposed to get to use on that cruise, and when they gave it to another team after promising it to him, he's like, oh yeah, you don't tell Walter no. So that's when he said, we'll build our own, and did it. Just incredible. So most of the funding was from the Smithsonian. So, you know, I think my salary was $200 a month plus room and board. Well, the room was really small, and the board was $1 a day per person for food. And so we ate a lot of rice and beans and caught fish, and I think his parents, who were from Newfoundland, they contributed money for the building of the ship, and then when the expedition was over, he sold the ship and paid his parents back. Okay, Mas.
Hi. So one of my favorite things to do is this triathlon in Monterey, where we get to swim through the kelp beds.
Nice.
Yeah, except now I'm feeling maybe kind of guilty. Like, what are we doing? Like, because there are, you know, I'm not the only one in the water at the time. So, like, how badly is that disrupting the life of what goes on in the kelp beds?
I don't think as long as you're not, you know, ripping kelp out off the rocks, I don't think it's disturbing a lot. I mean, the fish can dart away from you. The invertebrates, if you know, were happen to run into a kelp frond and knock some of the turban snails down to the seafloor, they might get eaten, and that's life. Or they might climb their way back up the frond again. I don't think you're bothering anybody, and you're doing such a good thing for your body.
In your years off the coast of California, what kind of changes have you seen to the environment due to climate change or other factors?
Changes in the environment off of California. We've been able to track changes in terms of, if you look at some of the studies that were done at Hopkins Marine Station in the 50s in the intertidal, where you have transects that are still marked, and then go back many years later and have them reassessed, you can see the species there are tending to be more of the southern species that you didn't see a lot of in the past, and the northern species are tending to not be so dominant. So you have some that are still there, that were always there, but there's been a shift. So the northern species are tending to maybe be not in Monterey Bay anymore, and more southern species are showing up. It was interesting, about 2 years ago, we went back to St. Croix, where we had spent a good year working up some of the data from Walter's expedition. And, you know, they've been hit so hard by hurricanes. They've just been beaten. But then the warming is also really delaying the recovery. I mean, we've always had hurricanes in the Caribbean, but these really intense ones, you know, especially if you have several in 1 year, it's been devastating to the reefs that were really beautiful. I would say, if you want to see a reef, get out there and see it now, because the future does not look very hopeful. Boy, that was a somber note. Any others?
It's so exciting to hear you talk. And I don't know if I'm more excited about outer space or landing in the water. It's just phenomenal. Anyway, I want to thank you so much. You're such a female role model, not just in the science, but just in general. Thank you a million.
Oh, What a sweet thing. You know, the reason I ended up going to Antarctica was because my friend, Joe Allen was an astronaut. And he was also on the Ambari Board of Directors, and I always tried to sit next to him at dinner. Just loved Joe Allen. And he came back from a trip to Antarctica with his son, and he said, you know, we never got along. And then we take this cruise to Antarctica on a Russian vessel where there are no rules. So if we